Hello, everyone, and welcome to this audio podcast. This audio podcast as part of the Calyx report edition. This audio podcast as part of a Bostix SandZtorm. This audio podcast as part of a squadron communique, and this audio podcast as part of a Storm Bostix production. In this audio podcast, a basis in around the pathogenesis, looking at nephrotoxic nephritis and glomerulonephritis, and sort of in around in around the basis about in around the basis around animal models versus human disease. So the pathogenesis of glomerulonephritis is generally complex. Intricacy arises from factors such as autoimmunity, cancer, and structural abnormalities within the kidney. There is variability in the susceptibility to GN, which is likely to have a genetic basis, and there are also complex interactions between soluble factors, including antibodies, complement chemokines, cytokines, and growth factors, and cells, for example, both leukocytes and intrinsic renal cells that mediate glomerular inflammation. The contribution of non-specific factors, such as proteinuria, for example, promote continuing renal damage or kidney damage, and despite resolution of an initial insult around the glomerula. A constant feature of GN is the presence of inflammatory leukocytes within the glomerular and interstitial compartments of the kidney. Macrophages and T lymphocytes are the dominant cell types present, and the intensity of cellular accumulation, particularly within the interstitium, rather than the glomerulus, has repeatedly been found in amongst the evidence to correlate with the clinical and pathological severity of disease in animal models and in human beings. So within the kidney, T cells and macrophages undergo activation, which is triggered by interactions between leukocytes and intrinsic renal cells, by interactions between cells, and a matrix, by cytokines and other soluble factors present within the inflammatory environment. Type 3 hypersensitivity occurs within the antigens, and antibodies are also present in roughly equal amounts, causing extensive cross-linking. It is characterized by soluble antigens that are not bound to cell surfaces in the case of type 2 hypersensitivity. However, when antigens bind to antibodies, large immune complexes from that cannot be cleared. The reaction can take hours, days, or even weeks to develop. Type 3 hypersensitivity represents sort of the progressive forms of glomerulonephritis or GN. That draws this audio podcast to a close. This audio podcast as part of a Calyx report edition. This audio podcast as part of a Bostix SandZtorm. This audio podcast as part of a Squadron Communique. And this audio podcast as part of a Storm Bostix production.